Good day to my supervisor, other reviewers, and my dear colleagues. Myself, Panchal Dias, and I will be presenting on the synthesis and characterization of porous copper aluminium ketone biosorbent hydrogel beads for effective removal of phosphate from wastewater. Now, passing through the overview towards the introduction, I have to mention water is one of the most crucial substances for the survival of plant as well as animal, and about 71% Earth's crust is covered with water, and yet the limited freshwater supply stands at about 2.5 from the entire water content. It is a source of water supply onto agriculture across countries, as well as it is very dependent on the countries supported with irrigation systems for general water supply as well as hydropower generation. Now, with urbanization and globalization, it has opened up and nurtured the birth of various production chains as per consumer needs. Countless different products such as cosmetics, fuels, fertilizers, detergents all send out effluents contaminating natural water bodies. According to UN, about 80% of the wastewater generated by all of these schemes are discharged into natural water bodies without treatment. And this study focuses on the chemical compound phosphate, which gets contaminated through all of such industries and removing it from the natural water. Aims and objectives are as such, where aim of this research is to conduct experimental work on developing different types of copper aluminium ketone biosorbent hydrogel beads, to conduct various testing to characterize and identify its effectiveness on phosphate removal. I've broken down this large aim into two objectives, where first I will conduct experimental research in preparation of this metal ketone hydrogel and with different ratios to compare and configure the optimum compositions, and that is to effectively remove phosphate. Now, second is to comply with the previous objective and to conduct various testing methods to verify the structure, anion absorption, and absorption performance with capacity over time. Now, moving along to the project's significance, three key points are highlighted here. This research focuses on achieving the UN Sustainable Development Goal number 6, as well as it is a guide for novel researchers and a reference for fellow researchers to conduct in-depth analysis on hydrogel bead incorporation in wastewater treatment. And lastly, it is also a reference for the UN SDG number 12, which is ensuring sustainable consumption and production patterns. Now, moving along to the most crucial part, why remove phosphate? Phosphorus is an element with a valency of 5, found as fluoroapatite, mostly abundant as a mineral rock deposit. It produces phosphoric acid when it comes to contamination with acidic substances. Now, passing through the naturally occurring mineral deposits, the natural phosphate cycle is crucial for all of us, inclusive of plants and animals. Now, the chemical phosphorus alone contributes to large part of the water pollution, and it is due to the three types of phosphate found on circulation, where they are typed as orthophosphate, polyphosphate, and organically bound phosphate. Now, phosphate also contributes into adverse effects ranging from eutrophication, ocean water acidification, and algae overgrowth in water systems. Apart from that, it can affect the human health by kidney disease or ethological dysfunction. Now, generally, a water body or a natural water body with a concentration of phosphate larger than 0.02 milligram per liter are classified as eutrophic. And natural national water quality standards for Malaysia, the recommended phosphate levels are at 0.1 and 0.2 milligrams for wet, fresh, and marine water respectively, where the health safety regulations of US but has mentioned that the globally accepted water phosphate levels for water would be around 0.05 milligram per liter. Now, moving on to the applications of phosphate removal currently in the existence, particularly the basic three categories are physical, chemical, biological. Under physical, the most common applications are ion exchange, magnetic field method, as well as electric coagulation. When it comes to chemical treatment, it is quite simple. It is the precipitation method using a coagulant. The most common types are aluminium chromate, the 3-valent ion as a coagulant, as well as calcium hydroxide. Now, biological methods give the best removal percentages according to the current existing treatment methods, and it depends from like fast treat to anaerobic oxic absorption and biomass absorption. And moving on to why use hydrogel beads. Actually, what are hydrogel beads? Hydrogel beads are cross-linked polymers that has a hydrophilic trait, and when once immersed in aqueous solution, it tends to attract water. It can usually swell several times of its dry weight and can keep the structure together when it's cross-linked chemically or physically. The component must usually account for at least 10% of the total weight to be called as a hydrogel as well. Three different types: resins, nanocomposites, and film. I am studying about resins, that is beads. And hydrogels are employed in various industries due to its unique architecture. Now, it is applied in various industrial to biological applications like drug delivery, dye removal, scaffolds in tissue engineering, and even contact lenses. So, moving on to the materials I'm using in this project, I'm using chitosan, which is a polysaccharide having a biocompatibility, biodegradability, and non-toxicity. Copper nitrate, why? Because copper is a reddish brown metal, which is a cubic crystalline structure, and it has a low chemical reactivity, and it falls into the category of transition metals. Most importantly, in hydrogen solutions, it is selected because it has the affinity towards phosphate. And when it comes to aluminium, aluminium chloride is chosen here just to increase the copper and phosphate attractions by aluminium focusing on the hydrophobicity. Given that aluminium is particularly not that strong of a metal, it makes sense when it is used as an alloy with copper. It is light but robust. But the introduction of aluminium actually improves the hydrophobicity. Cellulose, on the other hand, is one of the mostly distributed organic polymers in the nature, and the incorporation here is crucial to ensure cross-linking inside the breeze, freeing copper to actually attract more phosphate more than bounding itself to chitosan. Emerging nanomaterials called as naturally generated cellulose nanofibrils are with high strength, a large surface area, as well as variable surface chemistry. That is why cellulose nanofibers are used here just as an enhancer to increase the hydrophobicity of the nanocellulose. And these are some general applications, which were some good research papers to read more on the application of hydrogel in wastewater treatment. This is the rough methodology. I'll brief it down to you all so that it won't waste any of our time. Now, first it is to prepare a chitosan sample and then segment it into various portions to prepare the hydrogel mixtures. Reminding on the numerical values, it is going to be a 2% chitosan solution diluted in a 2% acetic acid solution. Now, when it comes to the hydrogel mixtures, I will need to focus on the next slide to describe how it is going. So types A to I are the considerates of types. And these types are again developed in three categories: control, inclusive of microcrystalline cellulose, and inclusive of cellulose nanofiber. The proportions and the percentages are drafted here. And after the preparation of hydrogel mixtures, what is left is to prepare the hydrogel beads. That is done by incorporating a 2% sodium hydroxide solution and by suspending the beads for about a good 24 hours to ensure cross-linking. Once the beads are prepared, you wash them in DI water till the pH is reached at 7, and then we'll dry them for about good 4 hours at 60 Celsius to ensure all the water in the hydrogel beads are gone out. And these are a couple of pictures that I've taken during the lab experiments. And the results and discussion is comprised of these four categories. Quickly moving on to FTIR. The FTIR results were obtained using a Perkin-Elmer spectrum. Now, the amide stretch, the CN stretch, and the bending owing to the amide stretching and the absorption due to CH stretching are visible around the wave numbers 3000. And CH bending is visible around 1300, as well as the characteristics for chitosan. Now, the CO skeletal stretch is typically abundant on polysaccharides around 1100, and here is slightly shifted to about 1000. And the asymmetric stretching again is visible quite closer to 1000, and the copper and aluminium absorption bands are alongside 700 to 400. And when it comes to absorbance, first the checking was done for water absorbance. The most water was absorbed by type C to F, and actually type F CNF, and the lowest was recorded for type A control, incorporating only copper. And this is the most important part where I did the batch absorption studies for all the bead types, all 21 bead types, and at the end of first set, the results were tested in 12 hours. 5 types of bead reached 50% removal. After 24 hours, 13 samples reached 50. 7 samples showed 60% removal efficiency. And at the end of 48 hours, 10 samples recorded a successful removal efficiency of 60%. And the most outstanding bead type was type A at the end, where it scored about 80% removal. And the kinetic study was apparently done for the selected type A control bead, and when the temperature was set for about a lower value, 15 Celsius, it didn't produce any good phosphate absorption, stood for about 6.7 only. When the temperature was increased, the standing stood very average, around 66%, but still the room temperature of the best results. The batch absorption study was done using various number of beads in 3 different equimolar amounts of sodium dihydrogen phosphate, and when using 10 beads at the end of 12 hours, it was 66%. Increasing 5 more beads, it was increased up to 70% removal, and actually the absorption cap was reached in 12 hours this time when 20 beads were used. Now, sonicator here was used when preparing the CNF solutions, where I had to prepare a CNF pulp to introduce it into the hydrogel beads. Next is the FTIR machine, the pH meter, and the comparison between unsoaked and soaked beads. Now, moving along to the recommendation, in this research, several areas of improvement can be observed when analyzing. First and foremost, the composition confirmation was only done using FTIR analysis. There are several other testing methods to confirm a healthy microstructure, namely X-ray diffraction, scanning electron microscopy, and even thermogrammetric analysis. Discussing about the absorption of pH variance, it was not done due to time constraint, and that is lacking in this experimentation research as well. High molecular weight chitosan was used from A to I control, and low molecular when the synthesis of types A to F of MCC and CNF came. This could have highly affected the absorptive properties since the beads had less number of hydroxy and amine functional groups and van der Waals bonds to interact from copper to other additives. And the resulted anion titration might have had fluctuation in results due to parallax error, which can be improved by repeating the number of titrations played. And when it comes to the last part, the conclusion, in conclusion, this experiment was a very good success considering that 10 types of bead had phosphate removal percentage higher than 60. From the FTIR results, favorable bondings from copper to chitosan, chitosan to aluminium were observed, as well as introduction of cellulose made the rigidness of the beads increase. However, the enhancers did not show any affinity towards phosphate ion surpassing the set of control bead set, but type A control was successfully eliminating 80% of the phosphate in all samples. So the samples had temperature fluctuations showed still the numbers are good at room temperature, and lastly 20 beads in 12 hours can get the job done, which in the first experiment was completed in 48 hours. So scan for references. That's all from me. Thank you very much.